Hello, everyone, and welcome to our webinar, Unlocking the Future of Vision, Cell and Gene Therapy in Ophthalmology. I'm Dr. David Tanzer, the Chief Medical Officer at Lexitas, and I'll be your moderator for today's session. We're thrilled to have you join us for this important discussion of how cell and gene therapies are shaping the future of ophthalmology. Over the next 35 to 40 minutes, we'll explore the framework, regulatory, scientific innovations, and future trends that are driving the space forward. We have an outstanding panel of experts who will bring unique perspectives from the regulatory, scientific, and clinical development points of view. Now, rather than lengthy bios, I'll ask each panelist to briefly introduce themselves and share their role in advancing cell and gene therapy in ophthalmology. Zoe, let's start with you, then over to Ruby, and then to Dan. Zoe?
Thank you, David, for the opportunity to be here. It's very nice to be able to speak on the subject of IBCs and IRBs. My name's Zoe Worthington, and I work with Univo. They're a provider of central IRBs and externally administered IBCs. I've been working in the regulatory space with IBCs since 2015, ultimately as an IBC chair. And prior to that, I worked as a bench researcher starting at the University of Maryland in the field of microbiology and molecular genetics. So the graduation to regulatory field and hopefully making a difference to individuals on a more forward front was really where I was aiming to go. So thanks for the opportunity to be here.
My name is Tsai. I am the founder and president and chief scientific officer of Applied StemCell. So our company specializes in gene engineering as well as induced pluripotent stem cell technologies, and we support cell gene therapy using our gene engineering and stem cell based technology.
Thank you, Dan.
Well, it's great to be with you today, and thank you so much for the kind invitation. So my name is Dan Chung. I am the CMO for Beacon Therapeutics, pediatric ophthalmologist by training. Started in gene therapy back in 1994 as a fellow at the NIH in retinal gene therapy back in the day. Been in the space ever since. Was deeply involved with the development of Luxturna, which was the first gene therapy to be approved for a genetic disease by the FDA. And currently working at Beacon Therapeutics, we are a late-stage gene therapy company in the ocular space. And we are looking, hopefully, to bring another asset to patients in that space soon.
Thank you, and welcome all. So before we dive into questions, let me set the stage for the discussion. Cell and gene therapy represents one of the most transformative areas in ophthalmology, offering hope for conditions that were once considered untreatable. Today, we'll dive into what's working, what's changing, and what's next. And we'll begin with Zoe in terms of talking about the regulatory framework and oversight. We'll discuss IBC processes, IRB and IBC best practices, evolving regulations, and any NIH updates that Zoe wants to bring to us. So the first question for Zoe is, ethics and patient protection are at the heart of any clinical trial, but they take on added complexity in cell and gene therapy. Zoe, can you share with us what's unique in terms of considerations that sponsors should keep in mind?
Absolutely. So typically IRB panels tend to be a little more risk adverse when it comes to cell and gene therapy trials, as these tend to be a lot more novel than other studies that they may review. These types of investigational agents also have the potential to bring about permanent changes or have long-lasting effects. Often researchers don't really have a full understanding of the full potential of off-target effects or the transmission of changes in future generations. And so this is something that the IRB will consider quite deeply. And they do need to consider carefully the benefit balance compared to the scientific justification. The concept of therapeutic intervention versus enhancement does also pose potential ethical challenges for IRB review. Does the investigational agent treat a disease, or is it involved simply in improving traits? Another item that IRBs consider carefully is informed consent. Participants don't really often have a deep understanding of the treatment that they're going to be undergoing. Gene therapy often involves very complex concepts and terms such as gene editing, viral vectors, and they may not understand very clearly the the study itself or the possibility of any unforeseen effects of the gene therapy. So making informed consent very clear is really very key to success, I think, for IRB review. These types of trials often require long-term monitoring and also the use of acquired data associated with ethical considerations. So a participant may not be provided with the results of some or all of the genetic testing being conducted on the study. And there are ethical issues surrounding that. There are also privacy issues when it comes to both the participant and also their relatives, since there is a much higher chance of re-identifying a patient based on their genetic data, and it can often be very hard to dissociate the individual from the data itself. Other ethical issues that arise that we could also consider on the IRB side is the collection of samples or the data that's collected about sequences, particularly when it is in the context of whether that data is being reused or whether it's being commercialized. And then finally, I would think about equity in recruitment, especially when it comes to human gene therapy, as it's aimed at treating rare diseases, which by definition affects a much smaller portion of the population. And care really needs to be taken to ensure that these vulnerable populations, that's the rare disease community, aren't exploited, and that access to the treatments that are being discussed are really available to those individuals.
Thank you. Next question for Zoe. The regulatory pathway for these therapies is very different from traditional drugs or devices. Can you walk us through these distinctions and explain the IBC process versus an IRB review?
Absolutely. So, gene and cell therapy products do still require review by the IRB, and they're reviewed in a very similar manner to non-human gene therapies, but the IRB board would include a gene therapy expert. And then the key difference really is that there is an additional review required, and that's by an institutional biosafety committee or IBC. And this typically happens when the research or the institution receives funding from the National Institutes of Health. And this can be in any amount. It's not restricted by a certain amount. The review can happen at the same time as an IRB review, or it can happen before or after IRB review. They're not dependent. And in most cases, these studies do have to receive approval from both the IRB and the IBC before that research can be initiated. IBC review process begins in a similar manner to IRB review in that the site provides their central IRB or IBC with a protocol, an investigator's brochure, and pharmacy manual. But at that point, that's when the processes diverge. And this is because IBCs focus on biosafety at the site, where the research is being conducted rather than the ethics and the safety of the patient. The investigational product is followed from the moment it's received at the site until it's eventually removed from the site, either by a disposal as biohazardous waste, or whether it's destroyed or returned to the sponsor. The sites do provide details regarding where the investigational product is going to be handled at each stage. And by this, I mean when it's received, where it's stored, where it's prepared, where it's administered, and what happens to any waste from the study as well. There's also a site visit conducted. Here, the site visitor acts as the eyes and ears of the Institutional Biosafety Committee. They're really there to make sure that the IBC understands how that product is being handled and that the site is able to handle it in the way that they've described it. The IRB focuses on participant protections, whereas the IBC is really focused on the safety of the research staff, the close contacts of the participants, the community where the research is being conducted, and also the public health and the local environment. IBC review does cover a much narrower range of studies than IRB review, and the committee structure is different too, compared to an institutional review board. I think the key thing to consider is that the review is conducted at the site level, so it's very local and includes people from the local community on that committee. Studies always go to full committee review, and sites may be required to have additional safety equipment to be able to conduct research.
Wonderful. Thank you. The next question for Zoe is, sponsors often face delay during reviews. What best practices can help streamline IRB and IBC processes for ophthalmic trials?
Absolutely. So I would say probably the primary thing is getting your documentation to the IRB or the IBC as quickly as possible. So the protocol, the investigator's brochure, and the pharmacy manual. And then using a central IRB and also an external IBC can really streamline that process, particularly when it comes to multiple site studies. There's a consistency that comes across with using the same IRB and IBC at every one of your sites. And also the initial study can iron out issues that the committees may have with the study, and then subsequent sites can benefit from that information and get to review and approval much quicker. Using an external IBC when sites are only allowed to use their local IBC is helpful too, because the external IBC can help the site prepare for review by that local IBC and speed things up. Identifying sites early so that we are able to recruit members for the IBC rosters speeds things up, and preemptively registering rosters ahead of time, as there is a delay involved in registering your roster with the National Institutes of Health. And reviews can't start until there is an IBC roster in place. I would also say consider having your external IBC look at sites you're considering to see if they really are IBC ready, if they have the equipment they'll need for that particular investigational product. Making sure that all the documentation adequately describes the investigational agent, so things like viral vectors, whether they have had competency testing, generational information about the vector, the serotype, virulence, host range, and tropism. All those things will get your study reviewed quicker if the committee can understand the product and how it was made. Ensuring that the storage, preparation, administration parameters are clear in the documentation will speed things up. Making sure that you're detailing off-target effects and stability. And then specifically for ophthalmic studies, there's often inflammation concerns associated with the target tissues and the modes of delivery. So addressing that in your documentation, specifically the protocol and the informed consent, will definitely help speed things along.
Perfect. Thank you. Last question for you, Zoe. Transparency and reporting requirements have evolved significantly. What are the recent NIH changes that sponsors should be aware of, and how do they impact trial expectations?
So the NIH earlier this year made some more minor changes to increase transparency with regard to regulation of gene therapy and clinical trials. They made sure that now the membership of all the registered IBCs are published and they're available to the public on the NIH's website. This brings the possibility of greater public scrutinization, but also encouraging accountability with regard to making sure that the members of the committee are appropriately qualified, and also that there are no conflicts of interest with the site where the research is being conducted. Additionally, they required that meeting minutes are actually posted on the site's website. They are allowed to conduct some redaction to remove either proprietary information or personal information, but they must be posted on the website. And again, this brings up another level of accountability and transparency. Moving forward, the NIH is actually working through a process to further update the NIH guidelines and the way in which biosafety is being monitored and conducted within clinical trials. So right now, they're currently surveying biosafety professionals nationwide, and they do have plans, as I said, to update the guidelines in 2026. What I've heard from other biosafety professionals and documentation that the NIH has put out there is that they're looking to make it easier to conduct low-risk trials, with the possibility of maybe removing the requirement for IBC review. But then in addition, they're going to expand the types of trials that are going to require IBC review beyond, I think, just gene therapy. Clinical trials will also need to think about addressing biosafety issues more directly, be more transparent with regard to how the research is being conducted, and the steps that the institutions where the research is being conducted will actually take to ensure that public health is being protected, as well as the environment and the study participants.
Wonderful. Thank you, Zoe. Well, the next sets of questions will be directed to Ruby Tsai and discuss innovation and application, and include new technologies, translational challenges, and supporting documents and developments. So first question, Ruby, innovation is moving very fast in this space. What recent advances in stem cell or gene therapy stand out to you? And what lessons have we learned from what you've worked on and what hasn't worked?
Yeah, thank you, David. So cell gene therapy in general has shown very exciting outcomes. So for ophthalmology, we're seeing very meaningful progress in cell therapy, either with or without gene editing. For example, earlier this year, FDA approved the first cell therapy in Celto for rare eye disease that shows basically slows down retinal degeneration through the secretion of a neurotropic factor. So this is a gene-edited cell therapy, the first one that's approved by FDA. And we've also seen several cell therapy approaches in ophthalmology. For example, embryonic stem cell-derived RPE cells, retinal pigment epithelium cells, have demonstrated very good safety and some early visual improvements in AMD, age-related macular degeneration, and the Stargardt trials. And iPSC-derived RPE therapies pioneered by the Japanese group under Dr. Takahashi proved that gene-edited and well-characterized iPSC lines can be manufactured reproducibly and also implanted safely. Right? And we also seen encouraging results from retinal progenitor cells sponsored, for example, by Tsai. So on the other hand, there are clear examples of what has not worked. For example, unregulated stem cell clinic injections have shown severe adverse events due to poor characterized non-GMP cells, and also cell products engineered with random integration create gene expression silencing, genomic instability, and batch variability. So this really calls for a shift toward more precise, site-specific gene editing. For example, using CRISPR-Cas9 and also using our Applied Stem Cell's own proprietary technology called TARGET for site-specific large cargo insertion. So I would say overall, what has worked is a stable, well-engineered cell source, precise genome editing, and also strong QC and a delivery method that match to cell biology.
Thank you. Next question for you, Ruby. Moving from discovery to clinical application is a major hurdle. How is your organization helping sponsors bridge that gap?
Yeah, so as I mentioned, at Applied Stem Cell, we have technology platform that includes our proprietary gene editing technology and also our GMP grade iPSC cell banks that have a drug master file with the FDA. So we basically help sponsors advance gene edited iPSC derived therapies using our gene editing platform to enable clean, site-specific large cargo insertion up to basic 50 kb into the genome to create stable GMP iPSC lines, which can serve as foundation for RPE, photoreceptor, or retinal progenitor therapies. And also beyond iPSC, we support sponsors using other cell types, such as primary retinal cells, progenitor cells, such as neural or retinal progenitor cells. So we support cell therapy development from discovery stage through GMP manufacturing stage, including providing feasibility, engineering characterization studies, process development, and optimization tailored to the biology of each cell type. And of course, GMP manufacturing, including seed, master cell bank, working cell banks, and all the way through drug substances and product manufacturing. And so we also support strategies to satisfy regulatory requirements, right? So our goal is to really ensure that a sponsor starts with a very robust, reproducible, clinical aligned manufacturing platform.
Thank you. Well, speaking of manufacturing and delivery methods can make or break a clinical trial. So what key CMC and logistical factors like stability, distribution, site readiness, and scalability should sponsors address early to avoid delays?
Yeah, so David, you're absolutely right. So for ocular cell therapies, delivery strategy basically shapes CMC design and strategy, right? So for example, for subretinal delivery, so it usually requires small volume, but high viability preparations. And so this really makes stability, thought prep workflows, and site training very essential. Right? And for intravitreal delivery, usually used for mesenchymal stem cell, progenitor cells, and engineered cells, it really demands different potency and distribution considerations. And it requires large doses and sometimes repeated administrations. So from a manufacturing standpoint, scalability is a major early decision point. Right? So whether working with iPSC-derived RPE cells or MSC-like populations or other progenitor or primary cells, so sponsors must plan for scalable differentiation, expansion, while preserving phenotype, potency, and purity. And also robust analytics that can track critical attributes across scale changes. And stability data that support shipping, storage, and also multi-site clinical execution. And as you mentioned, Dave, so site readiness still is one of the most common sources of trial delays.
Thank you. And finally, for you, Ruby, for companies entering this space, the challenges can be daunting. What do you see as the biggest hurdles, and how can partners help overcome them?
Yeah, so I see the biggest hurdles are several, right? So they're biological complexity, CMC rigor, and also the fragmented nature of the ophthalmology clinical operations, which Lexitas is really addressing that problem. So I like to comment on CMC. So that's what Applied Stem Cells specialize in, right? So when it comes to gene edited or differentiated cells, consistent quality, batch to batch quality is essential. So how do we help? So at Applied Stem Cell, we provide clinically ready, gene editing ready, such as GMP grade, gene editing ready iPSC lines as stable starting materials. Right? So you start with a very stable cell source and has a drug master file with the FDA already. So your CMC part is pretty much covered. You just reference our drug master file. That really provide a shortcut to your product development. And also we have established differentiation and characterization tools across multiple cell types. So that's really our specialty, is to differentiate stem cell into different kinds of cell types, including retinal cells and photoreceptors or other cell types. Right? And also we have experience in process development, and we conduct comparability studies, and we provide regulatory support aligned with ophthalmology specific expectations. So we strive basically at Applied Stem Cell to collaborate with sponsors to accelerate development and reduce risks. So sponsors can focus their resources on therapeutic innovation and outcome.
Thank you, Ruby. And the last set of questions will go to Dan Chung, and we'll discuss looking ahead, trends and risks in cell and gene therapy. We'll talk about the industry outlook, endpoint innovations, and future potential. So Dan, the first question for you is, Luxturna was a landmark approval in ophthalmology and in medicine in general, the first FDA-approved gene therapy for an inherited retinal disease. How did that experience shape the field, and what are the biggest hurdles that you faced?
Yeah, obviously it was a great achievement for the field in general, that gene therapy indeed could be successful. I think at the beginning point, there were a lot of things that we didn't know, that we had to basically set the path forward. That was 8 years ago. And I think there were new things that we learned about trial design, endpoints, regulatory work, and then, of course, the whole idea of commercialization of a product that is technically one and done. So there were a lot of learnings from that. There were obviously a lot of challenges, the fact that it was a rare disease, making sure that you had a clinical study that was powered correctly, and then really looking at different endpoints. And in that case, the development of a novel endpoint that really looked at what patients' deficits were and how we could improve that, but how do you quantify that? So those are some of the things that we were able to show. Overall, it was the idea that you could do subretinal injections on a global scale, that skill was transferable. Of course, gene therapy in and of itself was successful. The idea that you could have a proper immunosuppression regimen to minimize inflammation, and then the whole process of commercialization. So these were all early learnings. And obviously, things don't stay static forever, and it has now been 8 years, and the field has definitely advanced in many ways. But we still don't have another gene therapy approved in the ocular space. So obviously, there are some challenges for going further.
Thank you. Well, speaking of going further, beyond inherited retinal diseases, what do you see are the greatest potential for gene-based innovation in ophthalmology?
Obviously, gene therapy can be readily applied to monogenic disease, and that's what most of the inherited retinal diseases are. They're monogenic diseases. And that makes gene augmentation, which is the most common form of gene therapy, quote unquote. Now we think of gene editing as a form of gene therapy, potentially antisense oligonucleotides, CRISPR. There are a lot of genetic manipulations now. But for gene augmentation, we do see that there's an expansion in looking at more common diseases, such as wet AMD. Several companies are looking to really modify the way we deliver anti-VEGF products. Instead of routine monthly or bi-monthly or every 4-month injections, of being able to do it once and having a steady state of the product being released and treatment for that patient. Work is also going into dry AMD. There are 2 products out right now that have been approved by the FDA, and they require monthly or bi-monthly injections. And this can obviously set somewhat of a burden for this elderly population to come into major centers to get injections. And so potentially having one treatment that would hopefully last a significant amount of time would be very helpful in that space. Additional work is going into other vascular diseases, such as diabetic retinopathy, where things are looking promising in some early phase trials. So some of the things that we have learned from that rare disease, the IRD space, are really being applied to the AMD space and other more common retinal diseases, which is really just a great way to expand the field.
Great. Great innovation in terms of therapeutic applications, but let's talk about delivery methods and how rapidly they're evolving. What innovations in delivery excite you the most right now?
One of the great things about the eye is that we have many different methods, surgical methods, that we can actually get to the cells that need transduction for gene therapy. Obviously, there's the tried and true, somewhat standard of care right now is a subretinal injection. This obviously requires the talent of a vitreoretinal surgeon, an operating theater, anesthesia many times. And so it is a relatively advanced skill in the vitreoretinal sphere of techniques. Now with that, some of the modifications currently are in the vector design, because the vector really is how we get that gene to a specific area. And intravitreal injections are starting to become a little more popular, but is limited by the biodistribution of the vectors that we currently have. But many, many groups are looking at how to make those vectors more conducive to an intravitreal injection. And this obviously can be done in the office. This doesn't require operating theater, and any retinal specialist could do some procedure like that. And then a newer one is suprachoroidal. And this really involves a catheter placed into the suprachoroidal space and the product being delivered there. One company is looking at this for wet AMD, and the results so far look very promising in that. And obviously this is less invasive, but I do believe that when it comes to gene therapy, this might be on a case-by-case basis as to what cells you're going for, what disease you're going for. So there's definitely a lot of innovation when it comes to delivery. And I think right now it's not just the physical delivery of the product, but it's how we optimize the serotype and the vector itself to be able to get to those cells with these different types of surgical applications.
Great, thank you. The last question for you, Dan, is endpoints are critical for trial success. How are ophthalmic endpoints changing to support gene therapy? And what should sponsors consider with selecting them right now?
So endpoints has always been a big question, and unfortunately or fortunately, the IRD space has been somewhat put in a place where they have to take the established endpoints within the ophthalmic space, and many of those did not come from the IRD space. They came from the glaucoma space or the AMD space or the diabetic retinopathy space, and then the attempt was to apply them to the IRD space, which has not always been that successful. As you know, most of the regulatory bodies right now look at a handful of them. One is best corrected visual acuity, needing at least 15 letters on an ETDRS chart. The EMA has hinted at some times that maybe 10 letters would be sufficient in that realm. Low luminance visual acuity has also come with, that's what we're using in our pivotal trial, and the FDA has agreed that that also is acceptable, but the same bar as BCVA, 15 letters. And then we've had a lot of conversations concerning full field sensitivity light threshold testing, or FST. And this has been approved as a secondary outcome, but we're still facing some challenges with some of the regulatory bodies as an acceptance for a primary endpoint. And you know that in the IRD space, a lot of the first issues that patients have is light sensitivity, is they're losing the peripheral rod cells. And because of that, at dim light, they have very low vision. So FST is something that we're still trying to bolster the case that it could be a good primary endpoint. And obviously there's a lot of discussion on that and various opinions on that as well. The other is microperimetry, or another form of perimetry. Microperimetry is a little more advanced, much more robust in the data that it can provide. FDA has also put this at a relatively high bar, where they're asking for 5 pre-selected points within the grid that average 7 decibels. This is a very high bar. It's been difficult to get to. FDA has said that mean microperimetry could be used as a secondary endpoint, and the EMA has hinted that that is acceptable as a primary endpoint. Obviously, companies or sponsors need to develop that as to what is clinically meaningful of things of that nature. So there definitely is movement on the endpoint space, and obviously mobility testing. Luxturna, the whole MLMT is somewhat of a double-edged sword, because it worked great for RP65 patients because it was a very early onset, very fast-moving disease. So within 1 year, you could definitely see a change over time. But in some of the slower-moving diseases, such as retinitis pigmentosa, mobility testing should be adapted to really fit that disease. So MLMT may not be the correct mobility test, and there are other mobility tests out there now that are virtual. So many groups are going after that, look very promising. But it's one of those things that one size doesn't fit all. and you really need to tailor the indication to what type of mobility test would be applicable, and something that you could see within the timeline that you have for the duration of the study. So obviously those are coming out. AI is also out there, and I think there are a lot of folks working on the AI field of how we can use different things in AI. Structural endpoints are also being thought of more. The ellipsoid zone has gotten quite a bit of favorable signals from regulatory agents. We always have learned and been taught by the regulatory agents in the US that if you can show the preservation of photoreceptors, that would be a viable outcome measure, however you want to present that. And obviously it's up to us, the community, to really deliver that data to regulatory bodies so that they would be agreeable to those endpoints.
Perfect. Thank you. Fantastic insights from all panelists. We really appreciate it. And before we wrap up, I'd love to hear if any of you have comments or thoughts on what your fellow panelists have shared, whether it's something you'd like to build on, add perspective to, or even offer a differing viewpoint. Whoever wants to go first, please.
So I actually would like to echo what Zoe mentioned a few times about, especially when it's related to gene therapy or gene editing therapy, is really because it's a new technology. So FDA is really looking at off-target, right? So when we're talking about genome engineering, so especially like using CRISPR-Cas9, so it's not just, we're not just looking at the targeted gene editing, but we're also looking at, what kind of side effect, right? So because by the technology itself actually has this weakness of off-target, right? So the sponsor developers really need to keep in mind, have very robust analytics to measure the off-target events, just to show the safety of the drug, right? Of course, you don't want to have these off-target events to happen, right? So that's a big safety concern. So alternative strategy is really important. So I don't know if you guys know that just very recent, ARPA-H called for a grant called Thrive for gene therapy for hereditary genetic disease. And one of the focus is to identify novel technologies that will really, have better technology than CRISPR-Cas9, for example, right? To really make very precise, targeted gene engineering without off-target events. So applied stem cells, so we're in this field, and so our gene engineering technology by nature, it has almost negligible off-target event. So we're very, actually, I just got the news today, and we're actually moving to the next step. So we submit a summary proposal. Now we're moving to the next step for this ARPA-H grant application. So, but the main point I have is really talking about when it comes down to gene editing and gene therapy. So some of the things that's very special, specific, is really off-target events that the sponsors, developers need to consider from the very beginning.
Thank you. Zoe, Dan?
Ruby mentioned something that I think is really paramount for success with certainly IBC review is training. And the nature of ophthalmic studies often involves lots of doses, repeated doses, sometimes high concentration due to volume limitations, and making sure that it's very clear in the documentation that how to administer these things and how to do things safely. That's really key for biosafety.
I just wanted to say one thing was that it's great to see all the innovations when it comes to genetic medicine within the space of the ophthalmology. I think the other thing too is that there were a few years where the interest in gene therapy, especially for rare disease, had waned a little bit with the unfortunate developments that some of the phase 3 trials did not meet their endpoint with that. But I would say in the recent year or so, we definitely have seen an increase in the number of successful funding rounds by biotech that are targeting rare inherited retinal diseases. And so we do feel that there is an upswing and a more positivity toward the field. And we're looking forward to a few more tailwinds coming up. So it seems like the pendulum is starting to swing in a more positive direction for the community.
Thank you all. So the final question for each of you is, if you could give one piece of advice to companies preparing to launch a cell or gene therapy program in ophthalmology today, what would it be? Zoe, I'll invite you to go first.
Thank you. From a regulatory point of view, I would say develop a good partnership with a central IRB and a reliable IBC that can help guide you through the early stages when you're going to trial to maximize your success. Partnership with the regulatory aspect, I think, really helps expedite things.
Thank you. Ruby?
Yeah, so my biggest advice from the CMC point of view is to build quality and scalability from day one. Because the science can be extraordinary, but if the cell line, genome engineering method, or analytic package isn't stable or well characterized, everything downstream becomes slower, riskier, and more expensive. Right? So it really considering or prioritize precise targeted engineering over random approach, for example, would ensure better efficacy and safety down the road. And engage your CMC and clinical teams early. So especially for ophthalmology, it's very unique because it's small doses, specialized surgical delivery, and very tight requirements, handling requirements. So bringing these operational considerations into your program design early will save you significant time.
And Dan?
So I think what I'll probably say is pretty obvious, and the idea is really look at the whole developmental package, not just early on. Look at the preclinical, obviously think about what a clinical trial will look like, and then what the regulatory space would. But beyond that is the payers, because in the end, the payers are the ones who are going to have to pay for it for patients to actually get the product. And so you really want to make sure that all your ideas are realistic at each one of those steps. And you want to bring in the expertise. If you don't have the expertise, whether it's through consulting or going to conferences or whatever, you really need to bring in that expertise. And then, of course, our guiding light in all this is the patient, and really how we're going to relieve the burden of this disease on that patient population. So that's the thing, is really have a holistic look. And then your timelines. It's always great to be aggressive, but be realistic also, because these are people and patients that are in these trials, and you can do everything right, and things still may take longer than you anticipated. So those are the things that I've learned, and I try to just let others know, is that look at the whole program from the idea, hypothesis, all the way to the end user, which is the patient, and everything in between.
I love that. It starts and ends with the patients. That's fantastic. Thank you. Well, in closing, thank you to our incredible panelists for sharing your insights and expertise. And thank you all for joining us today for this important conversation on the future of vision and the role of cell and gene therapy in ophthalmology. If you'd like to learn more or continue the discussion, please reach out to us at Lexitas. We're committed to supporting innovation and helping sponsors navigate this exciting and complex space. Until next time, stay connected and keep pushing the boundaries of what's possible in eye health. Thank you, and have a great day.